I'm going to be talking about a type of gene therapy called genome engineering and how this could be applied to efforts to cure HIV. Okay, it's very experimental, it's a new type of medicine, but I think you've heard from the previous speakers just the magnitude of the problem we're up against, and so I think it's time to consider even these sorts of approaches. So first of all, just some basic concepts that are going to sort of guide the talk. Obviously, you all know that HIV causes AIDS because primarily it destroys CD4 positive T cells, which are an essential component of the immune system. And gene therapists think about using genetic engineering to make the CD4 T cells resistant to HIV. And we hypothesize that this could have sort of two advantages. First of all, fewer HIV-susceptible cells should lead to lower overall levels of HIV in the body. But in addition, we also think that by allowing these cells to be preserved and not killed by HIV, we should go some way to producing an overall healthier immune system that can also fight back against HIV. And our ultimate goal is to have a sort of drug-free control of HIV, or what we refer to as a functional cure. So you may know about this story. There has been one case of an individual who's actually been cured of HIV, Timothy Ray Brown, the so-called Berlin patient, and he was cured because he received a bone marrow transplantation from a donor who had the genotype of being homozygous for a mutation, CCR5 delta 32, which leads to a defective form of the CCR5 molecule, and this is an essential co-receptor for most strains of HIV. So this is sort of showing schematically Timothy's transplantation, and if you like, overnight he went from having a CCR5 positive HIV susceptible immune system to taking on the characteristics of his donor and also becoming HIV resistant, and actually, to all intents and purposes, he is HIV free. The reason that this works is because hematopoietic stem cells, which are the stem cells that live in our bone marrow, produce all the components of your blood and immune system, including the CD4 positive T cells that are the major target cell and reservoir of HIV, but additionally other CD4 positive cells in the body, such as macrophages and possibly microglia, that could also harbor HIV. And so as in the sort of gene therapy community, we've long been thinking, is there a safer way to sort of mimic the therapy that Timothy received without having to undergo the sort of the rigor and the danger of a bone marrow transplant? And the idea behind this is that we would take a patient's own hematopoietic stem cells and make them CCR5 negative and then return them to that patient. So the way that my group has been working to do this is using genome engineering, which is a type of gene therapy based on the use of so-called targeted nucleases. And you can think of targeted nucleases as being sort of genetic scissors. They are engineered to recognize and bind to a specific DNA sequence, for example, the CCR5 gene. They then create a DNA break at that targeted site, and then subsequently, the host cell repair processes that tries to mend that DNA break can be exploited to achieve several precise genetic outcomes. Targeted nucleases, the three most common type are shown here on this slide. On the left, we have the protein-based systems, zinc finger nucleases that I'm going to be talking mostly about, and talons. In both of these cases, you have protein modules that can be engineered to recognize different bases of DNA, and that's how you achieve your specificity for your gene. And they are linked to an endonuclease that has been stolen from a restriction enzyme and that introduces the DNA break. And on the right-hand side, we have the newer and, if you like, more popular type of targeted nuclease, CRISPR-Cas9, which uses instead an RNA that's complementary to your DNA sequence to bring in the Cas9 protein, which in this case is the endonuclease. So all these targeted nucleases work in the same way. They introduce a DNA break at a specific site, but then the outcome of that DNA break can be in sort of two broad directions. First of all, if the DNA break is repaired by a natural process called non-homologous end joining, this is very imprecise, and can result in small insertions or deletions at the break site, which ultimately can lead to loss of gene function or gene knockout. Alternatively, and in the second part of my talk, I'm going to be talking about instead the more precise repair pathway, which is based on homologous recombination. And that can be usurped by introducing into the cell what we call a homology donor, which can act as a template and be copied to repair the DNA break. And at the same time, this gives us the possibility of introducing, for example, a single base pair change. So I will call that gene editing. Gene knockout is much simpler because it only requires the nuclease and not the homology donor. And so, of course, this led to the idea that we could use gene knockout to disrupt the CCR5 gene and in that way create resistant cells. So the first use of targeted nucleases to do this was by several groups, notably the University of Pennsylvania and sponsored by Sangamo Biosciences, to knock out the CCR5 gene using zinc finger nucleases. And the first cell product that was targeted were patients' own T cells. So the basic idea is shown in the schematic. Total T cells or enriched CD4 positive T cells are isolated from a patient. They're expanded ex vivo using very similar technologies that are now used to, for example, make sort of anti-cancer CAR T cells. And at the same time, the CCR5 gene is knocked out by introducing this specific zinc finger nucleases, and then the cells are returned to the patient, sometimes after the patient has received treatment with a drug that will temporarily reduce the number of T cells before the infusion. Okay, busy slide. Don't worry, I'm not going to go through it. This is just summarizing the 7 completed and ongoing clinical trials using this reagent, and more details are found in a recent review that's just come out in Blood. But to give you a high-level summary of the findings, these trials have shown to date that infusion of the zinc finger nuclease-treated ex vivo expanded T cells has generally been safe and well tolerated. The recipients have seen increases in both the total number of T cells as well as the specific CD4 subsets, and the CCR5 modified T cells in those populations have been shown to persist long-term in vivo. More interestingly, perhaps, are the last 2 bullet points, which are kind of some of the, I think, kind of intriguing findings from these trials. So first of all, in a cohort of chronically infected immunological non-responders, so these are people who are HIV infected, and even though they're taking antiretroviral therapy and they are able to suppress their virus, their CD4 T cells have not bounced back up to normal levels. So in these immunological non-responders, they actually did see an increase in their total CD4 T cells, and they also saw some decay in the latent reservoir, in a couple of cases up to an order of 3 logs. And then in addition, in a cohort of 9 individuals who intentionally stopped taking their antiretroviral therapy as a scheduled treatment interruption, 6 of these individuals showed some degree of viral control, in 2 cases at levels less than 1,000 viral copies per mil. So certainly not cured, but I would suggest evidence that this treatment has actually given them a renewed ability to start controlling their virus. So that was the results from the trials where patients' T cells have been treated, and my group has been working instead to develop these sort of analogous therapy, but in this case treating the hematopoietic stem cells. And the rationale is that by going into the stem cells, we may be able to have a much more long-lasting effect. Just to give you a summary of what we've developed, we worked with our biotech partners at Sangamo to optimize a method whereby we could efficiently knock out the CCR5 gene in human hematopoietic stem cells, and we did this by electroporation of the hematopoietic stem cells with messenger RNAs that code for the zinc finger nucleases. We also did a lot of preclinical safety and efficacy studies. In particular, we transplant the human hematopoietic stem cells into what are called humanized mice. The mice, if you like, grow a human immune system, and you can then use that to confirm that this manipulation of the hematopoietic stem cells had no impact on the hematopoietic potential. And we were also able to demonstrate that the CCR5 negative cells that we generated that went on to develop a CCR5 negative immune system in vivo could suppress HIV. And I'll just show you one piece of data. This is looking at the levels of HIV in the blood of mice, either receiving either control untreated human hematopoietic stem cells or the cells that have been manipulated with the zinc finger nucleases. And you can see the ability of the human immune system in the mice to control the virus. And at the same time, although there was an initial loss of human CD4 T cells, this was recovered in the mice as the virus levels went down. And we now have ongoing a phase 1 clinical trial, which is proceeding at City of Hope, recruiting at 3 different sites. And we've actually just started enrolling and treating the first patients. Now, because this is a CCR5 directed therapy, one of the exclusion criteria for this trial are patients who also have viruses that can use alternate co-receptors, in particular CXCR4. So this tells you straight away that we're going to need something else beyond just CCR5 knockout. So one of the things that we're working on now is whether or not we can use genome engineering in this sort of other part of the pathway, whether we can use homology-directed repair to make some very specific changes in human genes to confer new properties. And these candidate host genes that we're thinking of editing can be grouped into 2 broad categories, either dependency factors, which basically means the host genes that the virus needs. And of course, a very good example of that is CCR5 itself, where this variant basically confers resistance to HIV. But today I'm going to talk a little bit about the opposite type of host factor, which are called restriction factors. And these are actually host genes that the virus has learned to avoid. They are innate intracellular defenses. They recognize different aspects of the HIV life cycle. They inhibit the life cycle, and they can also, in some cases, trigger an antiviral state and an interferon response. So there are many of these factors that have been identified, a lot of them published in Journal of Virology over the years. Up at the top, I'm just highlighting probably the 3 most common, which are ApoBec3G, TRIM5-alpha, and BST2 or tetherin. And if you think about it, although I'm telling you that these factors inhibit HIV, by definition, any successful human viral pathogen such as HIV has actually had to figure out a workaround to get around the human versions of these proteins. And in the case of HIV, for example, it encodes a protein called VIF that degrades ApoBec, a protein called VPU that degrades tetherin, and it's figured out how to make it, the viral capsid, resistant to being recognized by TRIM5-alpha by coating it with cyclophilin A as a sort of a decoy. So these genes also are kind of in an ongoing arms race with the virus. And although HIV is, if you like, winning the battle at present, it's an interesting idea to think about what could human evolution do in the future if we didn't have antiretroviral drugs and we had to figure out a way to fight HIV. And so as a gene therapist, we'd like to anticipate that and get ahead of human evolution and think about what the next move would be. Okay, so one of the ways we can do that is we can take advantage of the species specificity of restriction factors. So here I'm just showing you a cartoon of the different exons of tetherin, ApoBec, and TRIM5-alpha. And of course, the human variants of these proteins are not able to restrict HIV-1. But if you look at the orthologs in different primate species, we find some examples of the same genes in monkeys that are actually able to restrict HIV, including Tantalus monkeys and rhesus macaques. And so you can compare the human and the primate forms and figure out which amino acids might be critical for efficacy, and then think about making those changes in the human genes. So for example, this mutation in tetherin, this mutation in ApoBec3G or in TRIM5 alpha, which have been stolen from the primates, are able to convert the human form into now a form that recognizes HIV. And one of my favorites is this cute trick that Sudhi Mangabis have done, where the part of the TRIM5 alpha protein that recognizes viral capsids has been swapped out with cyclophilin A, and this gives it an ability to recognize the virus capsid. So if you like, we're going to be using this aspect of genome engineering to turn people into, whatever your favorite primate is. I have problems sometimes when I talk about that, and people don't even like GMO salmon. Okay, so how do we do that? Well, one of the first things we had to do was develop the tools. I've told you that our ability to deliver, for example, zinc finger nucleases and affect gene knockout is now pretty advanced and in clinical trials, but doing homology-directed repair is much more difficult, and we spent about 5 years trying to get this to work. But recently we published on a method that combines the zinc finger nucleases with delivery of the homology donor as a DNA genome of a small DNA virus, the adeno-associated virus, or AAV. And here I'm showing you just an example of how you can put a stretch of homology to the CCR5 gene that sort of spans the DNA break site introduced by the zinc finger nuclease, and the homologous recombination process will allow you to, for example, knock in a restriction site that we can then use as a tag. And one of the other things that's quite nice about AAV viruses is that they come in different flavors. They're kind of very much a sort of an M&M virus, and you can usually find a serotype, either natural or engineered, that will allow you to go into your cell of interest, in our case, the hematopoietic stem cells, where we found that AAV6 was particularly effective. So this is just one slide of data taken from the paper showing you the ability to insert either a very small point mutation and a restriction site, or to actually insert a much larger cassette, in this case GFP. And looking over here, you'll see that as we increase the amount of the donor, we increase the amount of homologous recombination and insertion of the XHO1 site, and at the same time, we reduce the extent of gene knockout, which is the competing process. And over here, you'll see that we get stable long-term expression of GFP in our hematopoietic stem cells, in this case at about 23% of the cells, but only in the presence of both the AAV donor and the zinc finger nuclease that produces the initial DNA break. Okay, so then back to our restriction factors, and can we use this technology to make these changes? So I'm going to show you just briefly where we are with doing this for tethering and also for TRIM5 alpha. So tethering, as its name suggests, acts by tethering newly produced HIV viruses to the cell surface and stopping them from being released. Normally, it's taken care of by VPU, but the T45I mutation prevents VPU doing this, and so it's a way to make a functional version of the human protein. So what we'd like to do is introduce this mutation into the exon. But one of the problems we face straight away is, if you remember, the competing process is going to be this gene knockout, and the last thing I actually want to do is to take a perfectly good restriction factor and make it defective. So our strategy is, instead of introducing our DNA break close to the site where we want to introduce the mutation, we've also been looking at whether we can do sort of editing at a distance by introducing the DNA break into an intron, where hopefully any indels that may occur will be less damaging. So here's just some data showing you how, for example, in this case, we are using CRISPR-Cas9, and we have a panel of guide RNAs that introduce DNA breaks close to the desired mutation site or further away in an intron. They all cut at approximately the same level, but conventional wisdom has it that getting homologous recombination to occur at a much more distant site, up to 200 bases away, is expected to be more challenging. However, we found that using the system that we use, that is not necessarily the case. So here's just showing you the ability of the different CRISPRs to introduce DNA breaks. And now in the middle here, I'm showing you our ability to actually make the mutation we want to. These are duplicate experiments. And you'll see that even as we get further away with our potentially safer strategies, we are still able to get about 5 to 10% of the cells containing this mutation. And this is also occurs when we knock in the GFP cassette at that site. Just briefly with the TRIM5 alpha, normally the HIV core protects itself from TRIM5 alpha by coating itself with cyclophilin A. And so the two strategies we are taking are to either make a mutation which now allows the TRIM5 alpha to coat the incoming core, or doing the Sooty-Mangaby trick of knocking in cyclophilin at this site to make an entirely new human gene, TRIM-CYP, which is able to compete with cyclophilin A and now bind to the capsid. And where we are with that approach, again, we've identified in this case CRISPRs that can introduce DNA breaks either at the restrictions, at the point where we would like to make the mutation, or at a distance. And because we'd also like to knock in the cyclophilin A cassette at this site, I will just show you the data with a surrogate, which is GFP, showing that for all these different CRISPR guides that we've generated, we're actually able to get about 30% precise insertion of GFP, which I think suggests we're going to have no problem doing that with the cyclophilin. Okay, so then in the last couple of minutes, I'll just touch on one last thing, which is the idea that we could also, instead of just knocking out a gene like CCR5, we could instead think of using this technology to inactivate an integrated latent HIV genome. And I will just preface this by saying that although, a lot of people have sort of suggested this, it's going to be a huge challenge to deliver something like a zinc finger nuclease against HIV to the very rare but also huge number of cells that contain these integrated genomes. And I haven't figured out how to overcome that problem yet. However, along the way, it's relatively straightforward to design targeted nucleases that can recognize HIV. We go in particular to the most conserved parts of the genome. That kind of makes sense. And showing that they work in a cell line is quite straightforward. Here's a cell line containing an integrated latent HIV that when we activate it becomes green. If we treat the cell line with, in this case, it's a talin that recognizes the HIV LTR and then activate the cells, we now see almost complete loss of activation. So at least in a cell line model, these targeted nucleases are highly effective. To try and get a little bit closer to what might happen in vivo, we've switched to using a humanized mouse model. Again, if you remember, humanized mice have grown a human immune system, and as a result, you can infect them with HIV. Here's the levels of HIV in 4 different animals. You can start them on antiretroviral therapy. We use a regimen that we incorporate into their food, and you can see suppression of the virus, and then if we change their feed back to regular food, the mice are happy. I don't think they like the drugs all that much, but the virus comes roaring back, and you'll see the same sort of things in untreated and ART-treated mice looking in lymph nodes and the thymus. You can see very similar things that are seen in monkey models. And so we use this mouse model to quantitate the latent reservoir, and we do that by using a strain of HIV that contains a cell surface marker. In brief, we harvest lymphoid tissues, such as the spleen, from the mice, and it's a mixture of cells, some of which are uninfected, some of which will be latently infected, and some of which will be productively infected. we can deplete the productively infected cells by virtue of the fact they express this marker. In this schematic, it's the red cells. And then we end up with a population of uninfected and latently infected cells. And we can look at the extent of the latent reservoir by stimulating the cells, in this case with a sledgehammer, CD3 and CD28, which will reactivate latently infected cells. And we can see that if we measure the levels of virus in the supernatant, and also if we do in situ hybridization to look for RNA. So that's the model. We can then take our anti-HIV talons and treat this population of cells with the talons prior to doing the stimulation. And now when we do that, we do see an ability of these talons to remove probably 80 to 90% of the latent reservoir. This is the control cells. These are cells treated with an unrelated talon against CCR5, and these are the cells that were treated with the HIV LTR specific talon. So again, I think, it works, but the problem is going to be we're not going to be doing this ex vivo. We're going to be having to think of a way to deliver these in vivo. So to summarize, I've been telling you about how genome engineering tools can be used to engineer either the virus or the host. Zinc finger nucleases that disrupt CCR5 have been evaluated in several clinical trials using a patient's own T cells and now also hematopoietic stem cells. We've recently shown that combining targeted nucleases with AAV vectors to deliver so-called homology donors is allowing us to get high levels of precise gene editing in hematopoietic stem cells, and I believe this opens up the possibility of editing restriction factors, creating new anti-HIV genes, or precisely inserting anti-HIV genes at a specific locus. And then finally, I showed you some data with talons and CRISPRs that target highly conserved regions of the HIV genome, and clearly these can be used to disable virus replication and can prevent reactivation from latency from integrated genomes. But of course, considerable challenges remain about the delivery of such reagents. Now we'll just conclude with my acknowledgements slide, and particularly with my colleagues at Sangamo Biosciences and City of Hope, and the people who've funded the work. Thank you. I'm out of time, yeah.
One question?
If you like.
Paula,
Maybe a question.
Thank you for an excellent talk and for making a difficult subject easy.
Oh, thank you.
But I just wanted to ask about your techniques and like the use of CRISPR, for example, in tissue.
In tissues?
Yeah.
So you mean in vivo or in tissues ex vivo? So we are trying to deliver the reagents to tissues in vivo, both to try and disable any HIV that is already there, but also because even strategies to edit human genes, CCR5 or restriction factors, it would just be so much nicer if we could just inject them into people, and they were sort of smart vectors and they could find their way to the cells we want to target and engineer the genes in vivo. And of course that's a real sort of holy grail for the whole field of gene therapy. Our approach is based on using several different viral vectors, including AAV, because you can engineer the surface of this virus to give it properties that could result in sort of targeted delivery in vivo. And we're also using combinations of viral fusion proteins with targeting moieties such as single-chain antibodies and DARPins that can recognize features on the cells that we want to target, such as CD4, CCR5, and markers for stem cells. So we have quite a large project doing that. I think it's an extremely difficult thing to do, but I also feel that it's kind of important that at least some of the groups involved in this area of research try to do that. And I will say that one of the things that's quite nice about working on something as crazy and difficult and important as thinking of how to cure HIV is that there is an appetite from the funding agencies to fund what is actually very difficult and highly speculative research with probably very little chance of funding. And oh my gosh, I sound like I'm on my soapbox here, but I also think it behooves those of us working in this space to take on those difficult sorts of projects.